Hello everybody, today we're gonna look at DNA replication. So first thing about DNA, intuitively you would think I would have to know about DNA to understand this subject, and you'd be right because it's DNA replication. So DNA looks a bit like this. So there is a phosphate molecule over here like this, attached to a 5-carbon sugar called deoxyribose. So this is the backbone of the DNA, and right under it, on these carbons, the one at the very bottom, oh wait, I forgot this hydrogen, one at the very bottom, there is a nucleic acid base attached to it, one of the A, C, G, or T, each for adenine, cytosine, guanine, and thymine. So I don't know if I'm pronouncing them correctly. So I'm gonna draw A, and I'm gonna draw over here a C, and onto them there is another nucleic acid base attached to it. For A, it always is T, thymine, and for C, cytosine, it is always the guanine, the G. And right under it, this is kind of turned over like this. It is not mirrored, not flipped, not mirrored exactly how it is. It is kind of rolled over, so it's kind of the opposite, you know, so it becomes a bit like so it kind of looks a bit like this. So it is definitely not flipped or mirrored, it is turned over. So if you look at the carbon, we're gonna name the carbon 1, 2, 3, 4, and 5. So we're gonna call this end the 3 end, and the 3' end, whatever, this and this end, because this is the 5 part, we're gonna call this the 5 end, 5' end. I don't know if they're called for 5' ends or 5 ends, but I'm just gonna call it 5 end. And so if you look here, it's 1, 2, 3, 4, 5. So where it was 5 end on the top part, it is 3 end on the bottom. Well, depending on where you look at it from it might be 5, 3, and 3. It might be 5, 3, and 5, 3 like that. It might be 3, 5, and 3, 5 like this. But yeah, the thing is, It is definitely the opposite, top to bottom. If something is going from 3 end to the 5 end, on the bottom part, it is definitely going from the 5 end to the 3 end. And note that the phosphates over here, these things, and this thing, and also this thing, and 3 on the top also, the phosphates act like the phosphates are just phosphates. Phosphates are a molecule that has 4 ends that have the oxygens, and I'm pretty sure they kind of act on their own. So see how I drew the oxygen. Do I call this a oxygen ion? The negative oxygen on the top, and I drew this on the top too. But if it was kind of turned over, wouldn't it be on the downside? But no, phosphates act. Well, I'm pretty sure, because I saw this on the book I learned this from. I'm pretty sure that they kind of act on their own, and it doesn't matter which side is facing up or down. So yeah, and these phosphates become kind of important too. So now, some of the other basics of what I'm going to explain today. Well, this was the basics of the basics. So most basic thing about the thing I'm going to explain today. So now the basic of replicating the DNA. So we're gonna have the DNA go like this, and one DNA is made of 2 strands, the 3-5 strand and a 5-3 strand. It might be 5-3 strand or 2-3-5 strand. Anyways, there are 2 strands. The strands are the thing with the sugar and stuff. The ribose and the phosphates and stuff, with the one of each nucleotide attached to it. So DNA is made of 2 strands, and this thing goes on a lot, and it's kind of twisted over, so it looks like a double helix. It is a double helix, and the nucleotides, each AT and CG, have hydrogen bonds to each other. They are attracted to each other, and they have the reason is hydrogen bonds. Now, so this is a DNA. The DNA wants to replicate. How does it do that? Well, firstly, something, probably a protein, unwinds the DNA. So it is unwinded and cut in the middle. The hydrogen bonds are broken. So the nucleotides, or the nucleic acids, are gonna be a bit like this. And then, with help of some more enzymes and stuff, well, it's gonna be enzymes and proteins and stuff, because that's what cells use to do stuff. And now, near them, there are NTPs flying about. NTPs are nucleotide triphosphates. So different from ATPs. ATPs are different in a way that ATP is adenosine, which is a adenine with a ribose attached to it with 3 phosphates right next to it. So the 3 phosphate derive energy, and NTP is kind of similar, but it is a nucleic acid base with a deoxyribose. This thing, the deoxyribose over here, with now attached to it 3 phosphates. So on ATP, adenosine triphosphate derives energy with the phosphates. The phosphates have energy in them. When there are 3 of them, the phosphates don't like each other, so if one goes off, zoom, when there's energy. NTP has energy too because of the 3 phosphates. And then they are used to derive energy while replicating the DNA. So these NTPs fly over and are stuck onto the already existing nucleotide. So, well, if this is an A, this would be a T. If this is a C, this would be a G. If this is a G, it would be a C, and if it's a T, it would be an A. Whatever is attracted to it is put onto the already existing nucleic acid base. So it is stuck onto over here, and then the deoxyribose, the sugar, this part is on top of it, and there are 3 phosphates, these things, right next to it. And if you see here, DNA looks like there's the deoxyribose, and then there's the phosphate. So and then another deoxyribose comes in right next to it, right next to the phosphate. But over here there are 3 phosphates, so you gotta get rid of the 2 phosphates at the end. But phosphates with each other very closely, they don't really like each other. So, as I said on the ATP, that's why they have energy. And as the 2 phosphates fall off, or, well, you could call it cut off, but as they fall off or get cut off, they derive energy because they had energy in them. But yeah, they give out energy because of the 2 phosphates have fallen off. And that gives energy to the enzyme that is putting these together. These NTPs could get stuck onto the already existing nucleotides by themselves, but that would take too much time, so they need an enzyme's help. So this gives energy to the enzyme, which now goes on to the next one. I forgot that one. Goes on to the next one and puts another NTP on that one. So NTP is placed on the already existing nucleotide after the DNA is unwinded, and the 2 phosphates from the NTP is gonna fall off, and give energy to the enzyme, which can now do some more work on the next one. It keeps going on like this. And this giving out energy and stuff is quite important too. And the which way is the phosphates gonna go, that's quite important too, but I'm gonna explain that later on. So yeah, this is the basics of DNA replicating. So if I draw a cleaner one over here, these are the nucleotides, and newly synthesized the outer layers. These parts are now over here with the nucleotides inside. I mean, not the nucleotides, the nucleic acid bases. They're different. Nucleotides are the nucleic acid bases attached to the phosphates and the this deoxyribose, and yeah, and on the top of it, the same thing happens. Another protein is there, and now DNA is made. 2 DNA from the already existing single DNA. So yeah, like that, DNA was replicated. And now on to some of the more specific things. I'm gonna erase the board. And now DNA polymerases. So DNA polymerase. Note that there is a A's at the end of it, which means it is definitely a enzyme. Something with the ASE on the end has a very high chance that it is a enzyme in a cell or whatever. It's definitely a enzyme. And now, what DNA polymerase what they do is very important. So a bit earlier on, I said that if this is a unwinded DNA strand, here are the nucleic acid bases. Well, of course on the top too, I'm not going to draw those though. So I said there is a protein, and the protein puts the NTP onto the already existing nucleic acid base, already existing nucleotide, and that protein is DNA polymerase. It goes on, gets energy from it, goes on, puts another thing, and then goes on another, so and so on. Yeah, DNA polymerase, it is probably the most important enzyme of DNA replication. It is very important. So yeah, and there are multiple DNA polymerases. So what some of the main things are: in E. coli, the germ, the bacteria that lives in your guts, and is fast at replicating, and is used in a lot, and I mean a lot, of experiments in biology, molecular biology. if you start studying biology, you hear it, you're gonna hear E. coli a lot of times because it is used in so many experiments. And yeah, in E. coli there is a DNA polymerase called Polymerase 3, and the 3 written with 3 I's, Polymerase 3. And in human cells and a lot of cellular beings have the DNA polymerase epsilon, DNA polymerase delta, I think, and DNA polymerase alpha. The DNA polymerase that does this thing is the DNA polymerase 3, DNA polymerase epsilon, and DNA polymerase delta, while the DNA polymerase alpha does something a bit different. I'm gonna explain that a bit more later too. So yeah, DNA polymerases are the most I don't know if it's the most important because the other enzymes are very important looking too, but yeah, very important enzyme. I'm gonna talk about a lot more proteins later on, a lot more enzymes. Enzymes would be the more correct way to talk about these because these are proteins that does stuff. And now, when DNA is replicating, oh, and I think I forgot to explain this at the basics, where I was explaining, and at the start where I was explaining about the things that you would have to know to understand these. I forgot to explain that DNA is a loop. I think it is always a loop, and even in chromosomes, the DNA inside our body and multicellular beings, the very long DNA strands that are sometimes kind of mashed up to form a thing that looks a bit like to form a thing that looks kind of like an X, like this. Even those are loops, and DNA is a loop. And when DNA is replicating, when that loop is replicating, the loop turns a bit like this. And well, because DNA is 2 strands, I'm gonna have to draw the strands, the 2 strands. Over here, it is stuck onto together, but over here, it is not stuck onto each other. It is, you can see, the space between it is more larger. And inside those, newly synthesized DNA are present. And another new strand over here like that. And this looks about right. The blue ones are the newly made ones, and the black ones are the already existing ones. So yeah, when this is replicating, you can see that there is a splitting part over here and another splitting part over here. We call those replication forks. In this replication forks, if I draw something over here, I drew it in the center. Why did I do that? And I'm going to draw a replication fork over here. So this is DNA, and this is split up over here. So this is the replication fork. Well, to be a bit more specific, if you say, a bit more kind of correctly, you might see the whole thing as a replication fork, where this is where the replication fork is starting. So a enzyme here that, well, as I said, DNA is all twisted up and is a double helix. The helix shape is all twisted back down, so it is not twisted, and then it is kind of cut open like this. When it was like this, all hydrogen bonded to each other. So the enzyme that turns the thing over, and so the enzyme that turns the twists the DNA in the opposite direction where it was originally twisted, and then opens up the DNA, is called helicase. The helicase works at amazing speeds of 10,000 RPM. So DNA is getting spun around at 10,000 RPM, rounds per minute, and opened up. So you can see DNA replication is quite fast. And on the nucleic acids, we have them over here like this. The nucleic acids, well, actually before that, I'm going to draw this a bit more down to be more symmetrical. That will make it easier to see. So the nucleic acids, the nucleic acids would want to stick back together because they are attracted to each other because of the hydrogen bonds. The hydrogen bonds want to appear, but there are proteins that stop it from happening, called single-stranded DNA binding proteins. So on these there are single-stranded DNA binding proteins. This stops from it binding onto together after the helicase did its job. So yeah, and then it falls right off before the DNA polymerase does its job, because I mean DNA does not have single-stranded DNA binding proteins inside of them, right in the middle. They don't look like that. These fall off right before the DNA polymerase does its job, and then they aren't needed anymore because DNA is made. it wouldn't want to stick back onto each other because there's a new strand on top of them. And now let's say DNA polymerases are over here like that. And also you have to know, I forgot to explain this also, but DNA polymerases, if this is a 3' and this is the 5', DNA polymerases always goes from 5 to 3. Very important. Always from 5 to 3. It does not go from 3 to 5, and for a good reason. And there's also something that helps it go from 5 to 3.
Hold up. Wait a minute. Something ain't right.
And I think I made a big mistake at the start and throughout the video. I said that DNA polymerases go from 5' to the 3' and make DNA going from the 3' to the 5', but it was the opposite. The polymerase goes from the 3' to the 5', while the DNA the polymerase is making goes from the 5' to the 3'. So now basically think of what you have learned in the video and kind of flip it. So kind of consciously think that, wait, that is the wrong direction. So yeah, just think of it as the opposite, and DNA polymerases go from the 3' to the 5', follow the 3' to the 5' way, and then make the DNA that goes from the 5' to the 3'. And using this information, let's say this is the 3' and this is the 5'. Helicase is doing its job and unwinds the DNA and at terrific speeds and going this way. But let's just say, let's just look at this situation relative to the helicase. So if you look at this relative in a frame of reference of the helicase, the DNA is coming at you. So the DNA is moving in this direction. So if the DNA polymerase is going this way, it is basically standing here while the DNA comes over here like this. So yeah, basically the DNA polymerase is going this way, but relative to the helicase, the DNA is passing by like this. And that is the correct way to go for the DNA polymerase, because it's 5 to 3, 5 to 3 going this way. So, but this over here is 5 and this strand is 3. This is a 3' and that's the 5'. But the polymerase has to go from 5' to the 3'. But the DNA is coming here like that and that. So how does DNA polymerase do its job on this strand for the 5 for this strand? How does it do it? Because it does not make any intuitive sense, but there is a kind of a genius solution to it. So the top part, I drew it as 3 to 5 like that. It's called the leading strand because there is no problems. There is no problems replicating the DNA here. It's just plain sailing, like zoom, and nothing happens much. But on the bottom one, I'm just gonna draw the bottom one over here. Actually, now we're gonna look at it relative to the DNA. We're looking at the same frame of reference of the DNAs, and so the DNA polymerase has to go from 5 to 3, so the 5' and 3', and the DNA polymerase replicates the DNA like this and goes back and polymerases and does its job over here, and then goes back again and does its job over here, replicates the DNA like that. So it replicates, goes back, replicates, and then goes back, and then replicates again, and then goes back, and this keeps on happening, and we call this the lagging strand. And each of these things, each of the kind of fragments that DNA is made, we call these Okazaki fragments because a person named Okazaki found how this works. Okazaki fragments make up the lagging strand, and so over here this is the leading strand, this is the lagging strand, and as I said, leading strand is just replicating the DNA without any problems, while the lagging strand is Okazaki fragment, jump over, another Okazaki fragment, jump over, and replicate again, jump over, replicate again. So yeah, it's a bit like that. And now also another very important aspect. When DNA polymerase does this job, it cannot start the replication job on its own. There has to be a RNA primer. So if it's going like that, well, this will be 5 and this will be 3. The DNA polymerase has to do its job, but it cannot start the job on its own. So if there is nothing on the DNA, it cannot do its job. But if there is a sequence, a short sequence of RNA this time, not DNA, RNA, called a primer, I'll write it here, primer. Oh, there's a YouTuber called Primer. Definitely check him out. He does cool science stuff. I like that channel. And this is our these primers are RNA, not DNA. Very important thing. So these little RNA primers are firstly made by an enzyme called primase. Firstly, there has to be an RNA primer made by the primase, and then the DNA polymerase sees that, this is a 5' and this is a 3', and the primer tells the DNA polymerase where to go. So, and then after the primer is made, the DNA polymerase does its job and makes the DNA right next to the polymerase. And now you would think, wait a second, if a RNA primase, if a RNA primer is right in the is inside the DNA that is replicated, wouldn't that be kind of bad? Well, wouldn't that be kind of weird to have RNA inside of DNA, which is definitely not DNA? RNA is not DNA. So, what happens to the RNA? And then, so there is a protein or an enzyme called RNase H. It's not RNase H because it has an A's in it. The A's at the end of it means it's a protein. The RN is ribonucleic, or is a ribonucleotide. Anyways, it's a protein. And then there is also a protein, exonuclease. And what exonucleases does, there are a lot of exonucleases. The exonucleases chop up the DNAs and stuff. If it's, it might be RNA or it might be DNA, but the exonuclease is a protein that cuts off things. It is, so the RNase H and exonuclease cuts off the RNA primer, and then DNA is, right after that made in the empty space, and that's how complete DNA is made. And now what about the lagging strand? At the start of every Okazaki fragments, there would have to be a RNA primer. So on the lagging strand, it is short RNA. The RNA primers are very short, like 3 to 10 nucleotides long. Very, very short. So the RNA, and then DNA, and then RNA, DNA, RNA, DNA. It goes on like that. The RNA primers are all then removed by RNase H and exonuclease. And then DNA comes in that empty space and completes the whole DNA. And now a bit more about a lot of proteins that are used in the whole thing. On the lagging strand, when the RNA is replaced by the DNA after the job of RNase H and exonuclease, there are a slight, gap between the DNAs. It is not yet put together yet. And then an enzyme called DNA ligase comes in, and then if the newly made DNA were about like this, you see the little gaps, right? These are glued together like glued together by DNA ligase. The DNA ligase puts the little thingy bobs together, the little fragments together. The DNA ligase glues the Okazaki fragments. And there are some more proteins that are used. I think I'm gonna have to erase this whole thing to explain it. Oh, and as I said at the start of the video, well, not exactly the start, but earlier on, when I was explaining about DNA polymerases, I said that a lot of polymerases are out there, and I talked about polymerase 3, polymerase epsilon, polymerase alpha, and how do you write delta? Is this something like this? It turns out it's this. So in E. coli, a prokaryote, if we have the leading strand, the polymerase 3 just does the job, while on the lagging strand, the primer is made by a RNA primase, and then the polymerase 3 does the job again. While in mammals, like us humans, the leading strand is polymerized by epsilon, and the lagging strand, the primer is made by primase, and the primase and the polymerase alpha, and the rest of it, the DNA part, is made by delta. And yeah, these are all the polymerases that I have talked about. There must be some sort of other polymerases and stuff out there. Now, about some more proteins. There are a lot of proteins used in DNA replication, and let's say this is a single strand of DNA that has been unwinded by the helicase from the original DNA, and there is a tubular sort of protein that is called a sliding clamp protein. In mammal cells in mammals, mammalian cells, is that right? Anyways, we have a protein for the sliding clamp protein. We have pCNA, and also, before that, what pCNA in mammals, or just sliding clamp proteins in general, what they do is they bind the DNA polymerase onto the DNA, so that if a DNA polymerase is binded here by the sliding clamp protein, over here would be over here there would be a newly pre-made strand of DNA by the DNA polymerase. The DNA polymerase is binded to the DNA by the sliding clamp protein, and the sliding clamp protein is, wait, let me draw a new one. The sliding clamp protein is binded to the DNA by a horrible drawing, but a clamp loading protein. So the clamp loading proteins, RFC in mammal cells, the clamp loading protein binds the sliding clamp protein onto the DNA, like this. If this is the single DNA strand, imagine it as a bit of a tube shape. I kind of get the picture. If this is a tube, there's the DNA going right through it. And yeah, binds the sliding clamp protein to the DNA, and the sliding clamp protein binds the DNA polymerase to the DNA. And now the DNA polymerase starts this job after the primer was made by the primase. And also one final protein, I think, called the topoisomerase. What topoisomerases do is, well, if this is the DNA, well, I would have to draw the swivels and stuff, but it takes too much time to draw, so let's just this is the replication forks. The helicases are doing the job of unwinding the DNA, but if the helicase spins the DNA at amazing speeds, wouldn't that lead to the DNA swiveling up like this? Because if you have a loop and you twist one side of the loop, it's gonna kind of twist up by itself because one part was spun very hard. So this has a high chance of, well, it's going to be bad for the DNA. Maybe the DNA would get all broken up, and that would be very bad. So DNA does not want that. So what there is a protein called topoisomerase. So let's say there's a topoisomerase over here. Then what the topoisomerase does is it kind of cuts one bit of the DNA, so that when the helicase does its job, it doesn't swivel up all the way like that. with the help of topoisomerase, even if it's the replication fork has been well, even if the helicase does the job even more, over here it's kind of cut by the topoisomerase. Even if time passes more and the helicase spins the things more and more, it does not get swiveled up like that. Because it was cut, even if it's spun a lot, it does not all swivel up. It's just fine. And yeah, that's what topoisomerase does. Another very important protein. If it weren't for the topoisomerases, I think, well, yeah, it would be bad. So now let's kind of summarize all the proteins. So now I'm gonna draw the DNA a bit like this, and this is the replication fork, and here is the helicase. So it moves this way, and about somewhere there's gonna be a topoisomerase which broke up the DNA so it doesn't swivel up. The DNA does not get all twisted and become a triple helix. Does it become a triple helix? Or helix squared? Now, anyways, here's the helicase. Spins up, unwinds the DNA, and let's say this is a leading strand, the left side. So right beneath the helicase, and here is, I'm just gonna draw it as two little leg thingy-me-bob parts, not among us. Now, this is the clamp loading protein right beneath the helicase, and there are little things that go out and kind of stuck onto the DNA polymerases, and here is the sliding clamp protein, and the DNA comes in like this, and because the replication fork is going up like that, over here is the newly synthesized DNA. And over here, this is the lagging strand. And wait, so it's gonna be 5' to 3'. Over here is gonna be 5' to 3', but over here, well, so over here, this is a lagging strand, and on the lagging strand, the DNA is a bit like this, and oh wait, I drew the polymerase and the sliding clamp protein on the wrong side. So over here is the sliding clamp protein and the polymerase over here like that. Now over here, the newly, wait, not that side. Over here, the newly made DNA strand like that. And the RNA primer over here. And over here, a primase is attached to the helicase, and the primer kind of goes up to the DNA and makes a little primer, and the primase goes up to the DNA and makes a little primer, and then falls off, waits for a bit, and then puts another primer there, falls off, waits for a bit, and keeps doing that. So let me draw here another primer, and the single stranded DNA binding proteins, they are like that basically on every nucleic acid before the DNA polymerases does their jobs. You can basically say that they're everywhere. And so this the Okazaki fragments, before they are made, the lagging strand part is kind of held up over here, and then the clamp binding protein puts the polymerase right onto the DNA strand, and well, most likely it will be over here. So then as the DNA passes by, well, actually this would be double stranded, and the DNA, the polymerase over here, and the DNA kind of passes by. So the DNA kind of goes like up, and so the polymerase can do its job and stuff. And well, there would be a the sliding clamp protein over there, and DNA goes up like that, and so the polymerase does its job and then comes over here. And then the thing is, the clamp binding protein takes the DNA polymerase off, and then the single-stranded DNA passes by through the sliding clamp protein, passes by, and then this protein, the clamp binding protein, puts the DNA polymerase back in where it was before. Actually, you know what? I don't think it was here before. I think I just explained some wrong information. It is always put here, but sometimes it is pushed all the way through the DNA. So if I draw it right here, this is the sliding clamp protein. It is pushed all the way through, and then the DNA polymerase is put onto it by the clamp binding protein, and then it is pushed up by DNA polymerases, by the DNA polymerase doing its job. It goes up while it gets replicated, and then the DNA polymerase is released, and the Okazaki fragment that has been made comes back goes back down, and so a bit like that, and then it is pushed down more by the helicase doing its job. So like that, this part now has to be replicated, and a bit more down there would be this part that has been pushed down, and then the polymerase is attached to it again, and then does its job, and this cycle keeps happening. This is what's happening on the lagging strand. All the proteins doing its job. It is kind of complicated, but kind of cool too. So it keeps on doing this. This is what happens on the lagging strand. The leading strand is just plain sailing, going and just synthesizing all the DNA. And yeah, that's about all the proteins that is there. Oh wait, I forgot about ligase. So, oh yeah, and the RNase H and the exonuclease, those, let's say they're about here, which removes the RNA primer, and then puts the DNA in it, so it becomes a complete thing made out of only DNA. And then a bit more down, there is a protein, ligase, more specifically DNA ligase. So the ligase puts the cut parts together of the DNA, because when the RNase H and the exonuclease did its job, and then a bit later DNA has been put in there, that part is a bit cut. Each Okazaki fragments are not put together yet, and the DNA ligase puts them together. And then when all the things have ended, the proteins now leave the DNA alone, and the DNA, and then there are two DNAs now. So the DNA has been successfully replicated. So now, when DNA is replicated, the most important part of replication, the actual replicating part by the DNA polymerase, when DNA polymerase does its job, basically, mistakes would happen. It just makes sense that mistakes happen, because I said that the nucleic acid bases put together, the force that puts them together is hydrogen bonds. But hydrogen bonds are just not strong enough. So the DNA polymerases just catalyzing the growth of DNA. It is just the hydrogen bonds are just not strong enough to make every little matches correct. Maybe there's a C, but an A comes in and tries to attach to the C, which would not happen. But the DNA polymerases, if they just simply catalyze the growth, not do anything else, but just put the things together, the nucleotides together, mistakes would happen. And if you look at it and do some stuff, calculations, it turns out that mistakes would happen almost every 100 nucleotides, which is a lot. Thinking of if you think about how many nucleotides are there, if the polymerase just catalyzed the growth, we would not be us. It wouldn't make sense that this much accuracy is in a replication of the DNA. So what happens? Oh, just saying that mistakes would happen if, a mistake would happen every 100 nucleotides put together, but actually, if you look at the statistics, we actually replicate with an accuracy of 1 mistake per 10 to the 9th power. That is about 1 per billion, I think, which is a lot better than 1 per 100. Billion is a big number, and so how does the accuracy, the worst accuracy, a very bad accuracy of 1 per 100, mistakes 1 per 100, to much better 1 per a billion? So now, firstly, there is a mechanism in DNA polymerase that does this. So, well, it just does not catalyze the growth. It actively checks if this is the right thing to come here. So let's see. A, C, G. Just a very simple, sequence. And so there comes T, G, C, A, T, G, C, but the polymerase over here. This is the polymerase, the circle, and let's see, somehow, because the hydrogen bond is a kind of a weak force, not A, but a G comes here, which would not stick to the T. The DNA polymerase sees that G is not the thing that would come here, and checks if G is the right thing to come here, but it's a T over here, so the G is not what should come here. So the polymerase is like, oh hell no, you go away. So the G goes away and the correct A comes here. And yeah, the polymerase just actively checks if this is the right thing to come. It has a mechanism of giving some signal to the polymerase if G or something that is trying to come here is the right thing to come here, checking the thing under it too. And this is a very important mechanism, lowering the percentage of lowering the chance of mistakes by 10 to the 5th power. So, which is 100,000. and 1 mistakes per 100,000. And then another very important mechanism, also in DNA polymerase. As I said, DNA polymerase is made out of a lot of proteins. It's a polymerase, so there are quite a bit of proteins inside it. So the part of exonuclease, DNA polymerase has its own exonuclease in it. Exonuclease, as I said, are proteins that cut off stuff, slices them up. So this exonuclease, well, let's say this is, the bottom one is the original strand, the thing that was already there, original DNA, and so it's just right till over here where on, let's say, a G has come. But G is definitely not what should come here. But the first mechanism of DNA polymerase could not catch the G from attaching here. The G has been attached, but the phosphates has not yet been fallen off. So, well, here's the DNA polymerase, and there is a part of, as I said, there is the exonuclease part. So, about here, the exonuclease goes from 3' to 5', opposite to the DNA polymerase, which goes from 5' to 3'. So the polymerase, the polymerase checks the bottom ones, while the exonuclease checks the top ones. But if it does not match, if the exonuclease finds a error, this strand, the top strand over here, is kind of turned over into the exonuclease, and as I said, exonuclease cuts off stuff. So the NTP of guanine is cut off, and the energy of cutting it off is derived from the 2 phosphates on the NTP. So exonucleases on DNA polymerases also get energy from the NTP. So this is cut off, and then the right thing will come and attach to the T, which would be an A, and then keep going, and then it would keep going on. Yeah, this mechanism is another very important thing, and this reduces the rate of mistakes about 100 to 1,000-fold, and so therefore about a mistake every billion nucleotides has been accomplished. But still there would be mistakes, and that's why mutations happen and evolution happened. But DNA replication tries to be as not wrong as possible, as accurate as possible. Oh, and I also said why primates is needed is to tell the DNA polymerase what way to go. That is also used here. If DNA polymerase was replicating from 3 to 5, from the 3 end to the 5 end. If it were to go from the 3 end to the 5 end, the exonuclease would have gone from the 5 end to the 3 end. But then if it went this way, the phosphates here would be on this side, which is on the side, if it were to go from 3 to 5, which is on the side where other nucleotides are. So the phosphate would have to be cut off before getting attached onto. But if this mistake happens, exonuclease would have to cut it off, but the phosphates are already gone, so the exonuclease wouldn't have enough energy to cut it off, and therefore the second mechanism that checks for mistakes, called proofreading, is would be not present, and there would be much more mistakes happening, which is a big problem. So that's why primers tell the DNA polymerase to go from 5 end to the 3 end, because of this, and because of the reason of proofreading. Yeah, and now about the origins of replication. I'm not talking about how DNA were made in the first place, of very early stages of Earth, before all this happened, all of the trees out there, me, you, and these bacteria floating about, the possibility of this virus over here, some prokaryotes living on this whiteboard, and everything. I'm not talking about before everything of this happened, how DNA was made. I'm talking about where DNA replication starts. So the origin of replication is, if we have DNA here, we have a special sequence of DNA base pairs that we call ORI, short for the origin. So let's call this place ORI, and then from ORI, when I say ORI, I'm not talking about Korean ducks. In Korea, we call ducks ORI, and I'm not talking about ducks, quack quack. No, this is in DNA, which is inside of ducks too, but in every other species too, so not just specifically talking about ducks. So now, ORI is a special base pair sequence. So in E. coli, we have found, people have found that in E. coli about 245 base sequences, base pair sequences are present as the ORI, which is a starting point of replication. And from the ORI, the replication happened. 2 helicases takes space and starts replicating with the polymerases and stuff. All the single-stranded DNA binding proteins and all the binding, clamp binding and sliding clamp proteins. And yeah, everything starts, and then there will be two DNA. Two DNAs that was replicated from the first, and now also there could be multiple ORIs per DNA multiplying, per DNA being replicated. So in E. coli is a very simple creature, a very simple prokaryote. So its genetic information ain't that long too. So E. coli only have one ORI, but us, for example, are very complicated. We have a lot of humans have a lot of genetic information. Even just talking about one chromosome, we still have too much. Even E. coli, only having one ORI, takes about 30 minutes to replicate itself, replicate its DNA, the genetic material. We are much more complicated. So if we were to replicate our chromosomes, well, if we just speak about one chromosome, if we were to do that, it would take about well, maybe multiple months to complete. Maybe not multiple months, but it would take too much to make just one cell, which is not good. We don't want that. If we get hurt, we would have to make skin cells very quickly. But if it takes like a month to make every cell, yeah, it's not gonna be good. So we and other multicellular beings, basically most multicellular beings and some unicellular beings also, maybe complicated beings, have multiple ORIs. So there might be one over here, one over there, replicating all. So multiple ORIs means faster replication, which is good. Faster the better. Human genomes, the human genome, the genome of Homo sapiens, have about 30,000 ORIs, the origin of replications. That is a lot. I'm talking about genomes here, the whole human genome, the 23 chromosomes, well, 23 sorts of chromosomes and 46 real chromosomes. So I think I'm talking about the whole 46 chromosomes here. So yeah, 30,000 for 46 chromosomes. I mean, that's a lot. 30,000 is a lot, where E. coli is a very simple prokaryote. It is simple, but it still is a living creature, having one ORI and takes 30 minutes to complicate, while we have 30,000 ORIs. That is a lot. And yeah, that's about all for ORIs. And Now about telomerases. Telomerase is another enzyme that is used in DNA replication. So, well, about lagging strands. Well, at some point the replication is gonna come to an end, but let's say this is the original DNA and this is the newly synthesized DNA, but it is this short. But this, let's say this is the lagging strand. There needs to be an RNA to proceed, but it is too short. Let's say it is too short for the RNA and the whole Okazaki fragment to fit in here. So this is where telomerases come in. So the telomerase comes in here like that, and telomerases have their own RNA fragments called telomerase RNA. So the RNA is kind of gonna be like that. So the end of the DNA, which is too short for replication on the lagging strand, this top part binds to the telomerase RNA, and it goes on for a bit, and on the RNA, temporary DNA takes place. So this part is temporary, but it is still made. So now the telomerase goes away, and it would be a bit like this, and then like that. And then this is, and then this space is large enough. It is long enough for the Okazaki fragment to fit in. So now RNA is made here by the primer, and then it is the DNA is made like that, and then the RNA falls off and DNA takes place, and the ligase puts everything together, and voila, you got a complete DNA. Well, of course, after that, the extra parts are gonna get cut off, or there would be mutations and stuff. You don't want mutations if you're the DNA. And then they're cut off, and everything is now well. Yeah, that's what telomerases does. It's quite simple. And yeah, I think that's about all for today, and all for this subject, and bye.